Today we are going to take a look at prokaryotic transcription. The goal of prokaryotic transcription is to synthesize a strand of RNA that will go on to translation. It is a necessary step for DNA to be converted into an enzyme that does a job for the organism. RNA polymerase is a protein that carries out prokaryotic transcription. In order for transcription to begin, the sigma factor must bind to the core enzyme of RNA polymerase. Once bound, RNA polymerase is known as the holoenzyme. Transcription begins after the promoter region, at +1. For this example, the top strand runs 5' to 3', while the bottom runs 3' to 5', since DNA is antiparallel. Within the promoter region, two consensus sequences aid with RNA polymerase finding the gene. These are at -10 and -35 on the promoter, relative to transcription beginning at the +1. Consensus sequences are observed to have extremely similar orders, suggesting there is some evolutionary advantage to having that specific order in that region. The RNA polymerase, as a holoenzyme, is able to recognize and loosely bind to the promoter region. Once the RNA polymerase is bound, it can start transcribing through elongation. Elongation is where the RNA begins to be synthesized. First, the sigma factor disengages from the RNA polymerase. RNA polymerase unwinds the DNA ahead of it and rewinds the DNA behind it as it travels along the sequence. The RNA is synthesized by adding RNTPs onto the 3' end of the growing strand. While the RNA runs 5' to 3', it is coding off of the 3' to 5' strand. This strand, the bottom one, in this example, is known as the template strand. The top strand of DNA is known as the coding strand. The RNA is complementary to the template strand and has the same base sequence as the coding strand, except for the exchange of uracil for thymine in the RNA. RNTPs are nucleoside triphosphates that have nitrogen and 3 phosphates. 2 of the phosphates are cleaved off for energy to form a phosphodiester bond with the exposed DNA using the remaining nitrogen and phosphate. Elongation continues until RNA polymerase reaches the terminator region. An inverted repeat sequence in this region, as well as the repeating A's at the end of the template strand, signal the end of transcription. This specific sequence allows the RNA strand to bind to itself and form a termination hairpin, which signals the end of transcription. The RNA polymerase disengages from the DNA. The AU bonds at the end of the region are weak enough for the newly formed RNA strand to separate from the template DNA. The RNA transcript continues on to be translated using ribosomes outside of the nucleus. Let's say the -10 box in the promoter gets deleted on a gene that codes for a sugar metabolizing enzyme. Since the RNA polymerase would not be able to identify the promoter region and begin transcription with the missing -10 box, the gene would not be transcribed and translated into a sugar metabolizing enzyme. The organism would likely die as a result of not having the enzyme necessary to metabolize sugar, since it would not be able to get the energy from whatever it consumes. And there we have an overview of prokaryotic transcription. Thank you for watching. Here are my works cited.